And these are results. The dark purple is the control, so the E. coli on each plate. These are the different treatments right here. And as you can see, this number 1, that's our control plate with E. coli on both sides of the plate. And you can see that it's pretty much 50-50, which is what we wanted to see. Okay, and 3 and 6, these were our potential pathogenic bacteria. And what we could see here is that they actually preferred the potential pathogenic bacteria instead of the control, which was very interesting to see. So also on the side, I was working with artificial soil microcosms, which have the intention of imitating their natural soil environments in a lab setting. And so I created our own protocol in the lab, and it included clay, sand, and vermiculite, and a variety of other smaller ingredients. And then also we put in natural soil bacteria, which is their food source, and then we added the worms in afterwards. And what I did was I tried to develop an optimum soil condition for their worm growth based on moisture level. So in the end, we came up with the mud-like texture was the optimum condition in which the worms survived up to 2 weeks or 2 generations. So in summary, I found strong variation in fertility and longevity on different wild bacterial isolates. I discovered preference behavior in worms, including strong preference for pathogenic bacteria. And I explored optimum living and growth conditions in artificial soil microcosms for future fertility, longevity, and food preference assays. So in the future, I'd like to examine the effects on fertility and longevity in the artificial soil microcosms instead of on plates in which they bathe in their own food, and it's not very natural at all. And then I'd also like to look at food preference within soil microcosms using gut content isolation. And I'd also like to isolate more bacterial species from the soil, since we only extracted 14, and in 1 gram of soil you can find thousands and thousands of different soil bacteria. So I would like to isolate more than just 14. So I'd like to thank everyone who has helped make this opportunity possible. ASC, University of Oregon, the Phillips Lab, and everyone in it. And Patrick Phillips, my mentor. Michelle Parmenter, who guided me through pretty much the whole summer. Anna Christ, who is also a very important person in the lab. Christopher Avon, who isolated the natural soil bacteria. Rose Reynolds, Jennifer Anderson, and the National Institute of Health, who funded the grant.
That preference thing was very, very interesting, I thought, especially because you got that weird result.
Yeah.
And I was wondering if there's, did you try varying how many bacteria that, was the assay sort of, do they chemotax to that side? Do they preferentially go to that way? And I was wondering whether these sneaky pathogens are putting out some perfume or something, that whether they're trying to mimic some nice one or something like that.
Yeah, that's what we were wondering too. We didn't go as far as to investigate that. That was just a preliminary assay, and we want to investigate that further in the future.
So it could be like if you put in a lower concentration or higher,
right,
some different smell would come out. Or
yeah, we'll try different conditions and stuff. Yeah. Any other questions?
When you say something is a pathogen, does that mean it's a human pathogen?
Not necessarily.
So what determines whether or not you call it a pathogenic bacteria?
Well, it depends on how the worms react to it mainly. As you can see with the fertility and the longevity, the potential pathogenic bacteria, they didn't really like them, and they died right away, and they did not reproduce that much. And from that kind of data, we can kind of determine that they are pathogens.
So they're pathogens for the, that's what
Right.
counts for the experiment.
So far that we
Right,
know,
yeah,
because
okay.
we just.
And I noticed in one of the early slides where you listed all the different bacteria that you had something about their genomes and a similarity index.
Mhm.
And could you relate that your findings at the end to what those showed, or did they not relate? Or were the closely related strains having the same effects on the worms?
Well, we found that similar bacteria that we had isolated out of the 14 were the same. Is that what you're asking? Yeah, there were like the same species and the same genome and stuff. So.
Okay, so
And they were similar results.
Similar
Yeah.
reactions
Yeah.
on the part of the
Yeah.
Yeah, I think this is very interesting. They are chemotactic, aren't they, in terms of finding it?
Yeah.
Any other questions? Yes.
Yeah.
What are possible practical applications of this?
You mean for like humans or?
Whatever.
Well, what we wanted to find, we just wanted to investigate more about the natural ecology of these worms because we still don't really know much about it. And so we used the natural soil bacteria that we had isolated, and that's just the first step in this whole process. So.